Since man's inception, they've always dreamt of creating something grander than themselves. Recent advancements in science and technology has given man the tools to do so, in the form of recombinant and chimeric DNA.
Chimeric DNA is a type of recombinant DNA, or more specifically, DNA containing certain genes derived from two or more species to create cells that contain genes of both organisms.
The origin of chimeras derives from ancient Greek mythology, from Homer's legendary epic, the Iliad. The chimera is depicted as having the back of a dragon, the center of a goat, and the front of a lion.
This is not necessarily what scientifically created chimeras are, but what they do exhibit are two or more genetically different cells. Chimeras can be created in multiple ways, such as through the transfer of organs, tissues, cells, or even DNA.
In the past 70 years, there have been major discoveries in DNA and its functions. This has opened the door for countless possibilities, including the birth of chimeras. Yet, as advancements have been made, those who oppose the creation of chimeras remain.
Paul Berg is a biochemist from Stanford University and studied connections between bacteriophages and tumor viruses that infect mammalian cells in correspondence to gene regulation. He began to investigate how mammalian viruses could take genes and move them to new cells. Due to the size of tumor viruses, an engineered virus was required for testing. The method discovered to achieve this required the splicing of two DNA molecules. DNA from a tumor virus and genes from a plasmid carrying E. coli were used. The gene splicing involved in his procedure involved splicing the genes in the DNA of the bacteriophage, or more specifically DNA of the lambdaphage. This is the first instance of the creation of recombinant DNA. Paul Berg was required to hold off his research due to the possible risks involved. Workers along with cultures of E. coli within the lab may be at risk if recombinant DNA consisting of tumor genes were to contaminate the rest of the E. coli within the laboratory.
Stanley N. Cohen and Herbert W. Boyer continue research into recombinant DNA and have contributed to the creation and understanding of it. Herbert W. Boyer was a graduate of Yale University. After university, he accepted a position at USFC. At USFC, Boyer and other biologists in the field found out that the enzyme eukaryote cut DNA and resulted in staggered ends.